Good day, dear students. Today we will learn about the process of DNA replication. What is DNA? DNA, deoxyribonucleic acid, is a long, complex molecule that carries the genetic information of all living organisms. It is the hereditary material found in the cells of all organisms, including animals, plants, bacteria, and viruses. DNA is composed of 4 different nucleotides, each containing a sugar, deoxyribose, a phosphate group, and a nitrogenous base. The 4 nitrogenous bases are adenine, A, guanine, G, cytosine, C, and thymine, T. The nucleotides are arranged in a double helix structure, with the sugar and phosphate forming the backbone of the molecule, and the nitrogenous bases forming the rungs of the ladder. The specific sequence of nucleotides in DNA determines the genetic information that is passed on from one generation to the next. The information encoded in DNA controls the development and function of all living organisms. What is DNA replication? DNA replication is the process by which a cell copies its DNA before cell division. During DNA replication, the 2 strands of the double helix are separated, and each strand serves as a template for the synthesis of a new complementary strand. The result is 2 identical copies of the original DNA molecule. The process of DNA replication involves several steps. The first step is the unwinding of the double helix by the enzyme helicase, which breaks the hydrogen bonds between the base pairs of the 2 strands. This creates a replication fork, where the 2 strands are separated and can be used as templates for the synthesis of new strands. Next, a group of enzymes called DNA polymerases add nucleotides to the growing strand, using the existing strand as a template. DNA polymerases can only add nucleotides in the 5' to 3' direction, so the new strand is synthesized in the opposite direction to the template strand. The synthesis of the new strand is discontinuous, occurring in short fragments called Okazaki fragments on the lagging strand. These fragments are later joined together by the enzyme DNA ligase. Once replication is complete, the 2 new DNA molecules each consist of 1 original strand and 1 newly synthesized strand. This process is called semi-conservative replication, as each new DNA molecule has 1 old strand and 1 new strand. Steps in DNA replication. DNA replication is a complex process that involves multiple steps, including initiation. The process begins when a specific sequence of nucleotides, called the origin of replication, is recognized by a group of proteins. These proteins bind to the DNA and create a replication bubble, which opens up the double helix. Unwinding. The enzyme helicase then unwinds the double helix by breaking the hydrogen bonds between the complementary base pairs, creating 2 single strands of DNA. Priming. The leading strand, which is synthesized continuously, is primed by an RNA primer, which is synthesized by the enzyme primase. The lagging strand, which is synthesized in short fragments, also requires RNA primers to initiate synthesis of each fragment. Elongation. The enzyme DNA polymerase adds nucleotides to the 3' end of the RNA primer in the 5' to 3' direction. The leading strand is synthesized continuously, while the lagging strand is synthesized in short fragments called Okazaki fragments. Proofreading. DNA polymerase also proofreads each nucleotide that is added, correcting any errors that occur during replication. Joining. The RNA primers on the lagging strand are removed by the enzyme NASH, and the resulting gaps are filled in by DNA polymerase. The Okazaki fragments are then joined together by the enzyme DNA ligase, forming a continuous strand of DNA. Termination. Replication is completed when the two replication forks meet each other at the termination site. The DNA is then fully replicated, with each strand serving as a template for the synthesis of a new complementary strand. These steps ensure that the DNA is accurately replicated and that any errors that occur are corrected before the cell divides.